I think we have a video that we wanted to show before I start. We wanted to show a video of Connor. We didn't get to show it yesterday of him walking, and so I'll just let it run. And for the people who weren't here yesterday, Connor was not able to walk independently before ASO treatment started, and so here you can see that he started to be able to take definitely more than one step, more than 2 or 3 steps, unsupported. And so this is what Kelly was talking about yesterday in her panel, and this is really, very exciting. This is not normal walking, but this is independent walking, and so I think this is very, very important. So we wanted to start out with that. All right, so now my turn to show you what we do in clinical development. I kind of feel like we have the easy job. I don't know, Nadina and Julie have really tricky part there, but we get to work with the investigators and then eventually the patients, so very blessed. You've seen this before, so this is where I fall within the processes of n-Lorem, but really I'll show you that the clinical team actually touches pretty much every aspect from case acceptance to patient treatment. I will focus on 4 main topics today, so I'll kind of be jumping around, so bear with me. First, I would like to introduce you to the clinical team, everybody that is helping me do what we do. I want to share a little bit more knowledge on the breadth of what we've been learning from the application, so kind of follow up on what Stan was showing yesterday. Then I want to show you how we've industrialized the clinical aspect and all the clinical processes to try to meet the needs of tomorrow. And then we'll go over some, clinical data to give you some more, understanding of the impact that we're having. But for the people who weren't here yesterday, and because, the clinical outcomes is really what's more important, I just wanted to give you a little snippet on, what we've been seeing. And some of this you've already heard in talks yesterday, but for the people who weren't there, just wanted to quickly summarize that in the patients that have been treated for long enough for us to be able to evaluate clinical benefit, we have seen clinical improvements. We have seen that for all the organs that we've been treating. And so in the central nervous system, we see increases at really low doses and very early in treatment, as was the case for Connor. We've seen it in multiple other domains, and Dr. Lisberg-Kravitz will talk about her case later on, and that will be a very interesting case as well. We see sustainable benefits in kidney function with our liver-targeting ASO, and Ryan yesterday was up here with Dr. Luang to talk about that. Eye treatment, we have an eye treatment. That patient has received multiple injections in both eyes, and that has been shown to be safe and seems to stabilize the disease. And so again, like Stan and everybody has said, we have really good safety and tolerability profile, which is really the most important thing for us. And so we see continued benefit, and I hope that next year I can come up here and show you even more benefit. All right, so now let's introduce you to the team. So obviously, the team is comprised of myself. I lead clinical development at n-Lorem. Joe Gleason, he's our chief medical officer. Cedric Gongyang is our medical geneticist. Helen Pugh is our clinical data scientist. And Stan is overseeing everything and providing input at every level. And so just wanted to show you that all of us are part of various different committees, either leading them or the lead person in those committees. And between all of us, we combine more than 50 years of experience in very different fields, from genetics to clinical trials to ASO expertise and ASO technology expertise. And so we're really well set up to be able to provide input at different points throughout the entire process of n-Lorem. And so just to give you an understanding of where we provide our clinical judgment, that is very important. So you've heard about the Access to Treatment Committee. So we help put the cases together, especially Cedric puts the cases together, and then we present those. And so we then present this to the team of experts that will help us make a decision on that. Once we start thinking about treatments, we work with the investigators to define the treatment goals, and we present those at the STAR meeting, and I'll go over some of those things in more detail in a little bit. In collaboration with Julie's team, the clinical team helps write various different sections of the IND, which will then be submitted to the FDA. Once the FDA has given us the green light to start a clinical trial, we work very closely with investigators to get IRB approval at their site, get the site set up, train the site on the protocol, and just make sure that everything's in place for the patient to be treated. And then, obviously, as treatment's going on, the Data Safety Monitoring Board, reviews all the data, and so we are the group that puts that data together for them to be reviewed. As Stan was also mentioning yesterday, it's very important for us to make the right decision on whether a patient, is amenable and suitable for being treated with an ASO. And so this falls in different categories. We look at the genotype, at the phenotype. There's also administrative things we need to make sure. Do we have an investigator, an institution that can, provide this? Because at the end of the day, we just want to make sure we can develop the right ASO, and give it to this patient in a very safe environment, an environment where we know we'll have clinical oversight. So specifically in terms of the Access to Treatment Committee, what the clinical team is doing there is, again, we're answering the questions, looking at the genotype and looking at the phenotype. With the research team, we're responsible to figuring out, is this mutation causal? So, if we try and fix this mutation, will it actually have the impact that we expect it will have? Is the mutation amenable to an ASO strategy? In terms of the phenotype, we really need to understand where the patient's at in their disease progression. As you know, these are nano-rare diseases, so we know less about the disease than we would in regular clinical trials, but we need to try and understand the manifestation of the disease. What is the prevalence? It's very hard sometimes to find out, how many patients in the world, or let alone in the US, are affected by this. We need to understand the rate of progression, because we want to make sure that we can assess changes in treatment, and we need to understand which organ is affected in these patients. So then, when we get to the point, once a case is accepted, like you've heard many times now, I feel like by the end of this, you guys will be able to come work at n-Lorem, because you will know the entire processes. But so by the time we're ready to start thinking about treatment, the case has been accepted. The clinical team works with the investigators to develop the treatment goals for the patients, because we will provide kind of like the blueprint of that to the Study, Treatment, and Assessment Review Committee. And again, together, we will try and determine if there's some assessments we've forgotten or not. And so it's very important that we really specify the treatment goals for this specific patient, to see what matters in that patient. And we also have to have assessments that are quantifiable, so that we can afterwards tell people and tell the FDA, see, this is working, and we have data to show. In terms of baseline data collection, we also need to make sure, like I said, that we know where the patient is right now. So can we assess change with using a certain assessment? And so oftentimes we ask the physician for the STAR committee to actually run the assessment on the patient before we present them to STAR, because we need to know, are they at the ceiling, are they at the floor, are they in the middle? Will we even see a change, right? And so all of that feeds into what we all have talked about is in terms of standardization of the assessments and how we collect these assessments, and I'll go into that in a little bit. Once treatment is ongoing, the clinical team, as soon as treatment starts, the clinical team reviews the safety data and the clinical efficacy data on a continuous basis. So this is of utmost importance to us. We need to make sure it is safe and nothing's happening to these patients who are already very sick. We also put a package together of all the data from our patients, and this is like individual safety data, but also aggregate safety data, and that is reviewed on a quarterly basis by our DSMB members. And the DSMB members really provides an independent review of our ASOs and an independent review of our processes. They also make sure that if we had to submit something to the FDA, we'll submit it in a timely manner and things like that. All right, so now that this is about what we do, I guess I can go off stage now. We're done, but no. This is what the clinical team does. But now I wanted to jump in and provide some more information on, some of the work that, information we looked at from the submission of the applications. And so Stan showed some of it yesterday, so this is part 2. For the people who weren't here yesterday, just to kind of give you an idea, between January 2020 and January 23, we've received 173 applications, so that's about 5 applications per month. A lot of those were for unique genes, which obviously makes sense, because all the applications that came in was basically a new gene until we got a few applications, for a repeat gene if people knew that somebody else had submitted something. If you look at the numbers from January 2023 to June 2024, we received an additional 123 applications. So now this has moved up to about 7 applications per month. And actually, the proportion of unique genes has declined, and we get more repeat genes in our applications, which again is very expected. People have heard that somebody with this mutation in this gene is being treated by n-Lorem, so now they're asking their physician to submit an application. So this is really not surprising. But what we have found is that in some of the newer unique genes that are being submitted is actually those genes seem to be more complicated. And so this makes for more complicated applications, where it just takes a little bit longer for us to go through it to make sure we understand the biology. Yesterday, again, Stan talked about some of the de novo mutations that we've seen in our patients. I wanted to kind of focus on, give you a couple of examples on mutations that are inherited, where we actually have accepted multiple patients from the same family. And so, and this really, familial mutations, as we all know, can lead to phenotypic diversity, but just wanted to give you two cases. So the first case is a family that has a mutation in SPTCL1. SPTCL1 is associated with both juvenile ALS and hereditary sensory neuropathy. And so here you can see the index patient is the father, who is 69. This is an autosomal dominant disease. And so all his children, the 3 daughters and the 1 son, are affected by the disease and obviously were diagnosed much earlier than the father. SPTCL1 is involved in sphingolipid metabolism, and perturbations in sphingolipid metabolism will lead to accumulation of toxic metabolites in the cells. What is also interesting in this metabolism and with these mutations is that if you have different levels of serine and alanine amino acid exposure, you can actually shift the phenotype from the hereditary sensory neuropathy to more of a juvenile ALS or ALS phenotype. And so this is exactly what we're seeing in these patients. And the father, compared to his children, has actually very different plasma lipid profile. And so this really underscores exactly what we're seeing here. It also raises interesting questions. Why is a mutation where they have the entire family has the exact same mutation? How come it really affects the metabolism this differently? And so it will be interesting to see, what we can expect in the future for these patients, what they can look forward to, but also how our treatment will be affecting the patients. We have actually also accepted another case. This is a non-related case of a patient who has an SPTCL1 mutation. The mutation is located really closely to that first mutation, so it's different, but it's also in the transmembrane domain. And that patient is younger and also has a phenotype that is more associated with juvenile ALS, which again is what has been known in the literature in terms of the involvement of sphingolipid metabolism and juvenile ALS. So the other gene I just wanted to touch upon quickly is mutations in DHDDS. So we have accepted 3 patients with mutations in that gene. 2 are siblings, and 1 is an unrelated patient. So DHDDS is involved in dolichol synthesis. Dolichol is really expressed ubiquitously, and so not surprising that it can wreak havoc in these patients. The first 2 patients have a mutation in the catalytic domain, whereas the 3rd patient has a mutation in the isopentyl diphosphate binding site. And so normally one would expect that mutations in the catalytic domain, which is a really highly conserved area, would actually lead to more severe phenotypes. But what has also been shown is that depending on the mutation and depending on that, whether it's in histine or glycine, depending on that change, it will actually change the conformational binding site at either the catalytic domain or the IPP. And so it can actually lead to even more severe phenotypes if you have that mutation in the IPP. And so in this case, if you just were to look at seizures, this is really kind of what we're seeing. So again, in this case, I think it will be very interesting to follow these patients once we have treatments for them to be able to see kind of like what we can do to help them. All right, moving gears again. Now we're going to focus on industrialization of what have we done in clinical development to make sure that we can, expand this and try to treat more patients, as best as we can. It's important at this point to realize that N-of-1 trials are unique, and so they require flexibility in thinking and paradigm. What we're really using in our trials is really a modified crossover design. A regular crossover design would be you treat with placebo, and then you treat with your drug. You may go back to placebo to kind of see what happens, on drug, off drug. We can't do that with our patients. Our patients are too sick. Antisense oligonucleotides last for a long time, so we can't do it. So what we're doing is modified crossover, where we will collect very robust pre-treatment data, and then we'll compare that to the post-treatment data on pre-specified goals. We also know that our patients are very ill and severely debilitated, they come into this clinical trial on multiple medications, they have different comorbidities, and they're at various stages of the disease. So we do not have a homogeneous type of population or, where you try to pick the patient that's just right for your clinical trial. We take everyone and we try to help everyone. We also have limited natural history data. So again, we know a lot less about the patients that we're treating than a commercial company would know about their patients. And while that all might sound negative, I just want to make sure people realize this is not a negative. This is really just something we need to acknowledge, and it gives us the opportunity to learn. And so like Stan said yesterday, we can learn from every patient, and learnings from one will apply to the next, and we'll make sure that we can share these learnings as much as we can through meetings like this, through publications, and so forth. So in terms of trying to scale this, right? So Sarah touched upon REDCap. So REDCap is an electronic database, and it captures the clinical data and the safety data that we collect. This is also a platform that academic sites are really familiar with. And so what we do is we have really focused on standardizing phenotypic data collection. And so these case report forms, they're basically forms that really lay out how we want the data collected depending on the assessment that you're doing. And so we're doing that with all the assessments that we're using in the trial, and we're doing that in terms of the safety, and so that we will be able to really compare from one patient to the next, and it's very simple for us to kind of look through this. By now, we have created a library of these CRFs, right? Case report forms. And so it's very easy for us to deploy the next database very quickly for the next patient. We can just take the books off the shelf and, patient 1 with phenotype X gets this, volumes, and then patient 2 with phenotype Y gets these case report forms. So this really, makes it very simple for us to, start up the treatment of a new patient. So this is a little bit to give you kind of an example of that. This is, looking at our pediatric neurology programs. So there's a list of assessments, that we have already, developed case report forms for. They focus on very specific phenotypes. So not every patient will use these in their clinical trial, but you'll see that, for example, focus on the ones that I covered here. Seizure diaries, a lot of our patients have seizures, so we use the same seizure diary, for all our trials, ask the patient to record it in the same manner. This will then help us, to obviously look within this patient what is happening with their seizures, but it will also give us some information of between a patient with an SCN2A mutation and a KIF1A mutation, did they respond to the seizures earlier in treatment, at what doses, and things like that. We, of course, also have patients who have very specific treatment goals, and that's what we were saying before. We developed these individualized treatment goals. And so we'll have patients who are the only ones who use a specific form right now. But I'm very sure that those forms, will be used in the future for the next 100, 200 patients that we'll be treating. The clinical group also obviously works very closely with the institutions. I think that's definitely our bread and butter, as working with our investigators. We are very excited that we have established really good relationships with a lot of institutions across the US. I call those our early adopters of the work that we do and of individualized medicine. So all the blue dots are different institutions that we're working with or treating patients at. It's great, but it's not good enough. There's a lot of white spots on that map, and we just want to make sure that we'll be able to interact with investigators, throughout the United States, so that the patients will not have to travel, too far, and that we'll have really excellent centers everywhere. This changed. So anyways, just wanted to show that the clinical team really works in close collaboration with the clinical sites from the beginning. And so at application, actually Cedric works with the site during the application very closely because he needs the investigators to provide additional information at times. But once a case has been accepted, we work with the investigators to receive patient cells, whole genome sequencing, because that is what Nadina needs to start the ASO discovery. And then we also work with the, with the investigators if they need to get institutional buy-in. Like we talked yesterday, not every institution jumps at this, right? And so we are very happy at n-Lorem to meet with everybody and anybody at an institution, if it is to explain to them what it is that we do and how this can be, absolutely awesome for their patients and even for the institution. During the ASO discovery, we obviously work with the investigators to, make sure that they start the baseline data collection. We track disease progression. we meet with the investigators very often and ask them, how's your patient doing? Has anything changed in their disease progression? Because if something has changed, and let's say they crashed, we may move this program up because we're like, okay, this patient cannot wait anymore. And then obviously we're working on developing the treatment goals during that time so that by the time we need to really finalize the protocol for submission to the FDA, we have everything set in place. And then obviously once the FDA has given us the green light to proceed, we will work with the site to make sure they get institutional review support, submission and approval. We develop the electronic database for them, and then we train the sites on how to run the protocol and things, basically things that any actually commercial company would do to set up a site and to make sure that everybody knows what they're doing once the patient comes in. All right, so now I just want to move on to quickly give you some more of the clinical update because we're very excited about that. But I first wanted to start with a slide that Stan has shown yesterday because this is of utmost importance to us. We first have to do no harm. And so, this is really like our guiding star. That is what's most important. Our patients are suffering from severe diseases. We do not need to add burden to these patients. They come in with many different medicines that have their own side effects. So we really need to make sure, that we're not adding anything bad. We have seen, just to say that, some patients have been able to get off some of these other medicines. And so, that has been very, very promising. But again, back to what Stan was saying last time, this really has not been done before. I mean, like Julie was explaining, this FDA guidance allows us to go into humans with a much reduced, GLP toxicology package. So we really need to pay attention to what happens and make sure that, we really look and track the patients closely. And so we feel that, for now, we need to be very, very cautious. But I think as we gain more experience, especially more experience maybe within one disease, then, we can become more assertive. All right, so this is our table of all the patients that are in active treatment right now. The green check mark on the safety means awesome safety. All right, so we haven't had any issues in all the 13 patients treated. As you can see, if you look at the first dose date, we have been treating more, 4 of the patients we're treating have been treated for more than 12 months. As everybody knows, Susanna has been on treatment for 24 months. We have, really great safety and tolerability. We have not had an ASO-related severe adverse event or adverse event. And we see clinical benefits in all of the patients that have been treated for long enough for us to be able to even look at clinical benefit. So there's improvements in multiple domains in the central nervous system at really low doses, and we keep on seeing continued benefit with time. So we have not reached a plateau in any of these patients yet. And also, as you saw yesterday in the session, or as you heard yesterday in the session with Ryan and Dr. Liang, we have target reduction in our systemic program, and it really seems to be stabilizing his renal function. So I just wanted to do a scorecard from last year to this year. And then I realized I was just extremely proud of this because we have not just doubled the number of patients we're treating since last October, but we have tripled the number of patients we're treating. And I know that between today, and actually I think one patient is being dosed tomorrow, but between today and the end of the year, we already have known dates of 8 more patients that will receive their first dose. And so we'll be in the 20s, right? We are treating multiple organs that are amenable to ASO strategy, and we're using several routes of administration. So I think, we now have a cumulative number of doses of 68. Stan mentioned yesterday that's 8 patient years, if anybody cares about patient years, but that just means the amount of time we've been treating patients for if you add all the patients together. So what is also exciting is, and people have touched upon this before, so these are the ASOs that are in the clinic right now, right? And so you can see which genes they, cover, and the ASO strategy. So as Nadina explained, some are a little selective. We really just had to focus to knock down the mutant RNA. Some are non-allele selective. It kind of knocks down everything. And then a couple of them are actually splicing ASOs. And so for some of these, we actually are treating patients or are about to treat patients, but we also have other patients we already accepted in our pipeline that will be able to use the same drug. And this is really actually exciting for the allele selective ones because, like Nadina explained, these are not designed to target the mutation, but they're designed to target a SNP. And so the new patient has to have the SNP on the right allele for this to even work. And so, I mean, it was a little bit amazing to us when we, looked at the whole genome sequencing and we're like, oh my God, this works. So, not only does this shorten the time to treatment for that patient, but it really also maximizes for us, the benefit in terms of, dollar per time invested per ASO. So will these be available to additional patients? Well, potentially. I mean, the non-allele selective ones for sure. Any person, should be able to use that, any patient. And then for the allele selective ones, it'll depend on the SNP. But again, we just need to look at the whole genome sequence and we'll know. So kind of just have a couple more slides, but just wanted to let you know we've really seen benefits across all our patients. And so the first set of benefits here are benefits that we've seen, using our quantitative measures like I was mentioning before, right? So we can see reduction in seizure frequency or seizure length. We have seen reduction or improvement in motor function and mobility, and these have all been tracked. Patients are becoming more attentive, have less repetitive behavior, less irritability. They are improving in their communication, whether it's verbal communication or communication using their device. And so it's really led to actually increased quality of life for the patient and for the caregivers. But then we've also seen benefits in some patients on things we were not necessarily collecting in a formal way. But we're learning now, and we will have formal ways to collect this in the future. But we've seen improvements in pain in Susanna. And we are lucky that families are recording this, so now we can go back to that. Improvement in drooling, increased stamina. So being able to stay in school all day, going to sleepaway camp for the first time ever. And then we've also seen improvement in the GI symptoms. I mean, this was with a regular scale that Kelly was using, but the scale could be improved in this case. But so I think it's really, very promising for us, all the changes we've seen, all the positive changes we've seen. So this slide really just basically says that. I think we're optimistic in everything that we're seeing, also because we have patients, in the pipeline. We have additional, targets in the eye in the pipeline. So whatever we see there in the first patient makes us very happy and optimistic for the next patient. The same thing with the kidney function. We're working on additional kidney and liver targets. And then again, in the CNS, it's just, in those patients, just been incredible. So just to conclude, I hope you feel that our clinical experience is outstanding to date. We have these processes in place to be able to scale this. So we will be able to treat 50 patients, 100 patients. I am completely sure of it. This is one that I'm very excited about because I feel like our nimble, experienced drug development team in partnership with the FDA has really been able to do this. And we are we are the ones who treat the most patients under this model. But of course, this could not happen without all of the pioneer patients. So I just wanted to take time here to thank the pioneer patients and their families who put the trust in us, because without you, we can't do what we do. So thank you to all these beautiful people, and that's it.